So this is your next video lecture regarding your lysosome, and today we will discuss about the functions of lysosome. Okay, um, let's get started now. Now, as the name of lysosome, we have already said that lysosome are mainly involved in digestion. Okay, so they are involved in digestion of what? Digestion of extracellular particles, digestion of extracellular particles, as well as it can also digest the intracellular particles also. Intracellular particles also, that leads to the formation of autophagic vacuoles that we normally know. Okay, now despite of this main function, they are also involved related with this function, they involve in other process also. The first function that we will see of lysosome is your endocytosis and the digestion of the macromolecules. Well, it is an important process that we have discussed during when we were talking about the functions of plasma membrane in our first cell biology classes. So the first function of lysosome is your endocytosis and digestion of your macromolecules. Well, the best studied function of lysosome is your digestion of your endocytosed macromolecules. From outside the cell, they are getting into inside and they are digested. Now, these molecules initially delivered into early endosomes, that macromolecules that we are talking about, they were initially delivered into early endosomes and then they will reach to the late endosomes before they finally reach to the lysosomes. So, in the process of endocytosis, uh, what we can say that first, if this is your cell membrane and there is some macromolecules surrounding extracellular environment, they are getting engulfed by the cell. So what I'm saying that first what happened, the molecules are first by the help of cell invagination, they are getting inserted into the cell, they are getting inserted into the cell in the form of endosome, in the form of endosome. These are the particles, the pink one. So this is called early endosome. This is called as early endosome. Early endosome, and then they reach to the final late endosome. They reach to the final late endosome. This is your late endosome, where this in late endosome will finally fuse with the lysosomes. So this is your late endosome. Then this late endosome will finally fuse with the lysosome. Okay, they finally fuse with the lysosome. So the hydrolysis starts from the endosomes, right? the hydrolysis of the macromolecules begins from the early lysosomes. They starts to degrade. The macromolecule starts to degrade, and here in early endosomes, the pH is having almostly about 6. Okay, and it is completed in the lysosome. Further, they will be mixed with the lysosome, where the pH is around, we all know, 5. Okay, a distinction. So here we can see that in endocytosis the macromolecules are digested by the cell, um, by forming, the cell forms an invagination to form an early endosomes, where the macromolecules are starts to be hydrolyzed, because in early endosomes the pH is around 6. So activation of the enzymes, acidic hydrolase enzymes starts to begin their digestion, and finally they form late endosomes, with finally they will fuse with the lysosomes having the pH of 5. Okay, normally the process of endocytosis and the process of your phagocytosis, if I say, uh, there is some distinction, although the process is almostly same, but the phagocytosis endocytosis process is a little bit different, because endocytosis process is common to all cells. This process of endocytosis is common to all the cells. All the cells can perform endocytosis, while phagocytosis is restricted to your specialized cell. If I say about phagocytosis, if I say about your phagocytosis, your phagocytosis process occurs in your macrophages. It occurs in macrophages, and it also occurs in neutrophils. Okay, so phagocytosis process is restricted to certain cells. Okay, they are found in phagocytic cells like macrophages, like neutrophils, right? Okay, so this is the first function that I have mentioned with you, that is your endocytosis. Okay, now we will do about your next function, which is your digestion of your external particles. Well, external particles means here uh, the particles from outside, foreign bodies, you can say, where endocytosis is limited to macromolecules which are found around our cells. Okay, but here for the digestion of extracellular particles, or you can say extracellular particles here as foreign particles here. Okay, like large particles, like your microorganisms. Okay, they are foreign bodies. Okay, they has to be degraded or digested by a process called phagocytosis. So we will write phagocytosis now. So the process of phagocytosis occurs in certain cells, like phagocytic cells, like you can say in case of macrophages, in case of neutrophils, this type of process occurs. These are the example of phagocytic cells. So large particles and microorganisms are taken into the cell by a process called phagocytosis. The cell actually engulf the whole particle, and then they form an invagination which gets pinched off from the cell membrane and form an internal sac inside the cell. So suppose if this is a cell membrane, and suppose this is the foreign body, let's suppose a bacterium. Okay, this is step 1. In the next step, what happened? In the next step, an invagination occurs. An invagination occurs. like this, and allows that foreign body to be engulfed, taken into the cell by the process called phagocytosis. And after forming that invagination, then what happened? They due to this invagination, the microorganism or the large particles get entrapped into that invagination, and it pinch off from the cell membrane and form an internal sac. Okay, it forms an internal sac. So what happens? It looks like this now. Invagination occurs, what we call as endosome here. Sorry, phagosome, not endosome. So this is known as phagosome. This we have studied in early classes. Okay. Now this phagosome will move towards the lysosome. It will move toward the lysosome, and there it fuses with the lysosome to form phagolysosome. So the lysosome, if I draw the lysosome in orange color, where all the digestive enzymes are present, so it fuses with the lysosome and forms phagolysosome, and the enzymes which are present in the lysosome, the enzymes which are present in the lysosome, they will bring about the digestion. Okay, so all the digestive enzymes, they started to break off the membranes of that bacterial cell here. Okay, and starts to degrade. Now the lysosome will not call as lysosome now when it fuses with the phagosome. It will now call as digestive vacuole, or at this position we will now call this lysosome as secondary lysosome. Okay, we'll call now as this as secondary lysosome. Now the residues, or the part left over in the digestion, are left in that digestive vacuole. Now this vacuole will move towards the cell membrane again. The part left over after this step, the part which will be left over will be now moving towards the cell membrane, and the process reverts to the phagocytosis, that is exocytosis, will occur. Okay, so if this process of if the process of digestion has to be continued, lysosome should be produced at the rate of phagocytosis. So here we can see that how the lysosome fuses with the phagosome and carries out the digestion of the foreign molecules. So if the process of digestion has to be continued in this way, then the rate of lysosome production should be similar with the rate of phagocytosis process. So this process of degradation of large particles generally occurs in specialized cells, like I have said, in macrophages, in your neutrophils. This type of phagocytosis event occurs, and these are the examples of phagocytes or phagocytous cells. Okay, so this is the second process of your function of lysosome. The third function is your digestion of your intracellular substance, what we call as autophagic vacuoles. These are the result of intracellular digestion by the lysosomes. So your autophagic vacuole occurs as a result of intracellular digestion. That means the cell is digesting its own internal parts or organelles. So one of the function of lysosome is your digestion of your substance of the same cell to which the lysosome belongs. So where the lysosome is residing, it is digesting its own cell parts. During the process, the substance to be digested will penetrate the cell's own lysosome, and this process is called autophagy. So in autophagy, what happened? Your lysosome, which is present in cells, this lysosome is this lysosome is I can say. This lysosome is actually allowing the penetration of the its own cell organelles actually where it is residing, and during that process the substance to be digested penetrates the cell's own lysosome. That's why it is called as autophagy. For instance, one best example of autophagy or formation of autophagic vacuoles occur in a tadpole tail, the young stages of frog. The tail of the tadpole generally disintegrates when it matures, and degradation of the tadpole tail occurs by the action of some enzymes. Okay, some proteolytic enzymes are there, and those proteolytic enzymes are called as catepsins. These catepsins degrade the tail of the tadpole when the tadpole becomes adult frog, when it matures to from adult frog. So we no longer use the tail, so it has to be degraded, and that is done by the catepsins proteins. And these catepsins proteins are found within the lysosome of the frog cell, right? Okay. Now the questions arise that what signals or what causes this type of proteins to carry out the autophagy. Okay, because autophagy does not occur continuously, it occurs at a certain time. What triggers or what stimulates this autophagy condition? How do large molecules get into the lysosome? These questions are very critical to be answered. It can be answered through research process. Okay, so here we have seen the third function of lysosome, that it can itself it can digest the intracellular substance the by a process called autophagy, and autophagy can be observed in many cases like degeneration of a tadpole tail when it matures to become adult frog, and it can be done by the help of some proteolytic enzymes like here catepsins. They degrade the tadpole tail, and these catepsins enzymes are found within the lysosomes. Okay, so this was about the third function of your lysosome. Then the fourth function is your extracellular digestion. Well, extracellular digestion I've already discussed, but it is somewhat reverts to your phagocytosis, where extracellularly they are digesting. That means the process of digestion occur outside. So in extracellular digestion, it is another process of a lysosome occurs in certain special cases. Okay, this means that outside the lysosome is carrying out its function of digesting the molecules. So this process is reverse to your phagocytosis, and what happens actually from the lysosomes, the enzymes that are found within the lysosomes, they get liberated outside, they get outside from the cell, and it digest the extracellular substance. One great example, best example to say this process is entry of the sperm. Entry of the sperm to the ovum occurs through this process. Well, sperm release some digestive enzymes with the help of the lysosome, which actually degrade the protective coating found in the ovum cell, and in this way sperm enters to the ovum to form a mature zygote. Okay, so this is another function, extracellular digestion. Now for digestion inside the cell, we know that any material to be digested has to form a vacuole, what we call as endosome. Then that endosome fuse with the lysosome to form a secondary, either with the primary lysosome or other secondary lysosome, and they brings about digestion. Then different kinds of hydrolysis, secondary lysosomes fuse, and the products of hydrolysis are passed out through the membrane or they can be utilized. Okay, now what happened? Resistant material then accumulates in the form of residual bodies. Sometimes what happen, undigested or incomplete digested particles may remain, and they may remain in the form of residual bodies, as I have mentioned in the first lecture of your lysosome. Okay, and this, and sometimes what happen in that we have seen in plant cells. In case of plant cells, we can see large vacuoles which contain the remaining parts of the macromolecules, remain after digestion by the lysosome. These are called as food vacuoles, which are found in the plant cell. So certain food vacuoles have been described in the plants, which are quite different from the lysosomes in the function. But that means it is opposite to the function of lysosome. Plant cells store nutrients in the form of food vacuoles, as I have said. About 30 to 90% of the plants and fungal vacuoles are occupied by the lysosomes, and as we know that lysosomes are the hydrolytic enzymes, but they also perform other functions within the plant cell like storage of nutrients and waste products like your proteins and other substance like anthocyanin in case of rubber plant rubber. It also degrade the waste products, right? So this is another function that we observed in the plant cells, right? Sometimes it also actually control the turgor pressure. By maintaining the H plus ions, by maintaining the H plus ions inside and outside the cell. Okay, so they have that transmembrane proteins, that pumps. With the help of active transport, they can transport that protons and maintain that intracellular concentration of H plus ions. Even the pH is also maintained by it, by them. Okay, so if I say the origin of your lysosome, actually, uh, normally lysosomes are exclusively formed from your Golgi vessels, and the hydrolases synthesized on the ribosomes as attached with the endoplasmic reticulum. So from the Golgi vesicles, your lysosomes are being formed, and the enzymes which are found inside the lysosomes, they are actually produced by the ribosomes found on the rough endoplasmic reticulum surface, which are passed out through the lumen of the endoplasmic reticulum, and then they different type of modifications occur like glycosylation. Okay, which are then transported to the Golgi apparatus. From there it will transmit to the pass on to the lysosomes. Okay, so this was all about the lysosomes. I hope you have understood this part. Just go through the notes that I have given, and I have tried to make it the notes here the lecture easy by writing some simple things here. So just go through it and just watch this video lecture, and for any doubts you can ask me in the WhatsApp group. Okay.